Potato cells contain the enzyme catalase. This enzyme catalyzes the breakdown of hydrogen peroxide into oxygen and water. Figure 15 shows what happened when a student placed a potato disc in 5% hydrogen peroxide solution. So what you've got here is catalase, which breaks down, it says here, breaks down hydrogen peroxide into oxygen and water. So what's happening here is this potato cell, which contains catalase, is breaking down this hydrogen peroxide solution into oxygen and water. The oxygen is making the disc less dense as the bubbles form on this disc, and it rises. The student measured the time taken for the potato disc to rise. The student repeated the investigation using 10%, 15% and 20% concentrations of hydrogen peroxide solution. So what we're doing here is we're varying the independent variable is the concentration of substrate, the thing we're breaking down. Which term describes the hydrogen peroxide in this reaction? Well, it would be substrate, which is the target molecule. The potato discs all had the same mass. Explain why the student used potato discs with the same mass. So you have to state the reason for that. So the mass is a control variable, okay, and that's because obviously potatoes contain the enzymes. So what you're controlling there is the enzyme concentration, because the only thing you're changing is the substrate concentration. So everything else which could affect the rate of reaction must be constant. So mass is a control variable. So there's an equal amount of catalase in each reaction, because obviously the potato discs need the same number of cells, which will have the same amount of catalase in them. Whenever you look at an enzyme practical, really there's only 4 things that they're going to vary. One is enzyme concentration, substrate concentration, temperature and pH. The one they're changing means all other 3 must be set or controlled. State 2 factors or other factors that need to be kept the same to improve this investigation. Well, I've just covered that ahead of time, so that would be temperature or pH. Figure 16 shows the result of this investigation. The student calculated the rate of reaction using the formula 1 divided by time in seconds. State and explain a conclusion based on these results. So what we're seeing, as the concentration increases, the independent variable, the concentration of the substrate, the time taken for the disc to rise gets lower and lower, and the rate of reaction increases. So in this question, we're being asked to state and explain a conclusion. So what is the conclusion, and justify why you think that. So the conclusion here, the obvious conclusion is that increasing substrate concentration or hydrogen peroxide solution, the concentration of it, will increase the rate of reaction. Increasing hydrogen peroxide concentration increases the rate of reaction, and that's 1 mark. Now we need to explain that. So basically, the more substrate there is, it increases the frequency of successful collisions between enzymes and substrate. So you can see here there's only a few, so as these are randomly moving around, there's less chance that they're going to collide successfully than when there's more of them. It's like the idea of dodgems in a ring. If there are more of them, they're more likely to collide, and then when there's lots of them, you're going to be colliding more frequently, but up to a point. Once all the active sites are occupied, then enzymes can only work so fast. So now enzyme concentration is the limiting factor. So we need to increase enzyme concentration to make this rise further. But here we're just explaining this bit, because all this is showing is a rise in rate of reaction. So here, so as it provides more substrate, 1 mark. So increasing hydrogen peroxide concentration increases the rate of reaction because it provides more substrate, which increases collisions, forming more enzyme substrate complexes. That's what these are here when they're bonded together, when the active site and the substrate are bonded together, releasing more oxygen. So that will explain why the rate of reaction increases, because oxygen is the product. Whenever you're explaining enzyme activity, just remember to talk about collisions, because there'll normally be a mark there. Use the terminology substrate, and use the idea of enzyme substrate complexes. This is a very fancy sounding word, but in every sort of explain question, you'll need to reference this. The student repeated the investigation with a 25% hydrogen peroxide solution and recorded a time of 75 seconds. So we've got 25%, 75 seconds. Calculate the rate of reaction for the 25% hydrogen peroxide solution. So if you remember that formula at the top, rate is 1 divided by time in seconds. So time is 75, so you would do 1 divided by 75, and we have 0.01333. Choose 3 significant fingers or 3 decimal places, whichever one suits. I would choose 3dp here, because if you look here, all the figures here are given in 3 decimal places, and you want to follow the same idea in your answer. So always look at the table and follow that as an example. So that's why I've done it as 3 decimal places here. Always check your calculations twice as well, just in case you made a mistake. Interestingly, the student decided that the rate for the 25% hydrogen peroxide solution was not anomalous. So if you look back here, we should expect, surely, with more concentration, that we get a rise in the rate of reaction, but here it's fallen down, it's gone down. But the student's saying this isn't anomalous, this isn't a mistake in the experiment, this isn't something that's gone wrong with my practical technique. Actually, this makes sense. Why does it make sense? Well, let's look back at this graph, because obviously if you keep increasing this substrate concentration, the amount of hydrogen peroxide, there's only so many active sites available to actually help or collide and break it down. So you can keep adding more, but if there's only this many enzymes, it's not going to speed up the rate of reaction, because these are all active sites are occupied. So that's why the rate is decreasing. We expect it to. So to answer this, you say substrate is not limiting the rate of reaction. All active sites are occupied, which is what this graph shows here.